Hi everyone, this presentation is about the biochemistry of DNA. We have already seen nucleotides, structure of nucleosides, and how glycosidic bond is formed, and how the phosphoester bonds and phosphodiester bonds are formed. Phosphodiester bonds between two nucleotides and phosphoester bond between a nucleoside and phosphoric acid. So this is about the deoxyribonucleic acid or DNA, especially the double-stranded DNA which are found in the eukaryotic nucleus. Okay. Now, this is a typical structure of DNA. Here you can see, as we have already seen, DNA, it is a polymer of deoxyribonucleotides, deoxyribonucleotides. And usually we have double-stranded DNA with two polynucleotides, two polymers or two chains of polymers of deoxyribonucleotides. You can see over here, this is one chain and this is the second one. Okay, and between them there is a bonding which is actually held between the two bases. Usually the bases, it is the bonding is between a pyrimidine base with a purine base, and even there is specificity that adenine always bonds with thymine and cytosine with guanine. Okay, now so this is how it is. Okay, you can see here this is the guanine and this is cytosine, this is a purine and this is a pyrimidine. Okay, and at three different points they form hydrogen bonds. Okay, and these hydrogen bonds, you can see the three bonds they bind guanine and cytosine. While between the adenine and thymine, they do have hydrogen bonds, but only two in number, right? So DNA contains double-stranded structure where the sugar and phosphate they form the backbone. Okay, you can see here it is found externally, right? While internally the bases are directed internally, and these corresponding bases are bound together by hydrogen bonds. So the structure of DNA is very specific. So how it was being elucidated and what was the basis for the such a kind of structure? So the whole structure of DNA, double-stranded DNA, it was proposed by James Watson and Francis Crick. I'm sorry, Crick, C has to be in capitals. So Watson and Crick model of DNA. It says that DNA contains two polynucleotide chains which are coiled in a helical fashion. You can see here this is a helical step case, and this is how the DNA strands are being placed. That is, there is a single axis in the center, and the two chains are wound around that particular axis. You can see here it's being wound around the axis. Okay, so somewhat like this, the two chains are coiled in a helical fashion around a particular axis, and that too in a right-handed helix fashion. Okay, so if you hold your right hand and fold your fingers, how the fingers are directed? It is the helical chains are wound around the axis in that particular direction. Okay, so that is how it is being arranged. Now, the backbone of the helix, it consists of sugar phosphate groups, alternating sugar and phosphate groups. So we can see there is a, what you call, the railing is there, isn't it? So this is actually made of sugar phosphate backbone. Sugar, then phosphate, then sugar again, then phosphate again. Okay, sugar and phosphate of one nucleotide, and then next sugar and phosphate of the next one. So there is an alternating sugar phosphate that is bound, that actually forms the backbone of the DNA double helix. And then the bases, the nitrogenous bases, they are perpendicular to the backbone and project inwards to the center. As you can see here, the steps of the staircase. These steps are actually formed by two bases. Okay, so the bases on the what you call nucleotide chain, it is directed inward. It is perpendicular to the helix. You can see here it is perpendicular to the backbone and it projects inwards. Okay, now the backbone, it is formed of sugar and phosphate, and it is they are hydrophilic in nature. They are hydrophilic molecules, and these hydrophilic backbones of the alternating deoxyribose and phosphate groups are present on the outside of the helix. That is, they face the external medium, which is actually water fill. Okay, or it contains water. Okay, so the hydrophilic backbone actually is in contact with the watery external medium, and hence the DNA is as such stabilized. Okay, while the purines and pyrimidines of both the strands, we have two strands and purines and pyrimidines attached to both the strands, they are stacked inside the double helix. I hope you got it clear. Okay, we have yeah, here you can see two helix. Okay, this is the backbone, and the pyrimidines and purines on both the strands. Okay, this is the purine and okay, two purines on this strand, the pyrimidines on this strand, they are directed towards the center. They are stacked inside the double helix with their hydrophobic ring structures very close together and perpendicular to the long axis. Okay, so that is very peculiar about how the sugar and phosphate is arranged and how the purines and pyrimidines are arranged. Okay, so this is James Watson and Francis Crick who proposed the structure of DNA. Okay, and yeah, so here you can see the basics, how the bases are being paired through hydrogen bonds and how the what you call the double helical structure, okay, or the two helix, the two chains, how it is wound around a central axis. This is central axis. Okay, so this is how it is being arranged. Okay, now we will see the detail of the structure. Uh, Here, the sugars are linked together by phosphodiester bonds between the 3' carbon and 5' carbon of the adjacent sugar rings. What does it mean? Is like that is two nucleotides when they are connected, they are linked. Two nucleotides are linked by phosphodiester bonds, and these phosphodiester bonds extend between 3' carbon of one nucleotide with the 5' carbon of the second nucleotide. So that is how the whole what do you call the backbone is formed. Okay, now DNA double helix, it is held together by two forces, hydrogen bonding between the complementary base pairs, right? So that is a pyrimidine of one chain, it is hydrogen bonded to the purine of the other chain. So that is a hydrogen bonding, and this actually hold the two helices together, or two like double helix together, to two chains together. Apart from that, we can also see base stacking interactions. Okay, the base stacked in the center, it shows interactions, and that also actually that is more important for holding the helix together. Okay, now a base pair, that is a purine and pyrimidine, consisting of a purine and pyrimidine, hence it is having a width of 20 angstrom. Okay, a purine and a pyrimidine structure, if you take the length, on an average it is 20 angstrom in length. Okay, from here till here. Okay, so that is the length, right? So each of these bonds may be differing, each of these structures may be differing, but on an average it is 20 angstrom with a single base pair if you take. Okay, A double bond with T and G triple bond with C. Okay, two complementary chains are anti-parallel. Why is it so? Because here you can see that the 5 prime, okay, this is a 3' 5' direction. As we have already mentioned in the previous presentation, this goes 3' 5' direction, and here it is just the opposite. 3' 5' direction is in this order. Okay, so here the picture gives 5', 3' in this direction and 5', 3' in the opposite direction, and that is why it is known as an anti-parallel arrangement. So the two complementary chains are anti-parallel to each other. Now, the ends of the molecule, they are asymmetric as because it is anti-parallel. 5' of one chain will face 3' of the other, and just the opposite. Okay. Now, the pitch of the structure being 34 angstrom and radius being 10 angstrom. Okay, we'll see the structure over here. Uh, so this is one what you call winding. Okay, so here you can see it starts from here and it has to come back over here. Okay, so this particular, this is a single winding. Okay, so this distance, it is 34 angstrom. Okay, and the width over here, it is 20 angstrom. So that is what is mentioned over here. Okay, the pitch of the structure being 34 angstrom and radius it is being 10 angstrom. What happens is two base pairs, two adjacent base pairs, they are just 3.4 angstrom apart. Okay, and now DNA helix, it is about 20 angstrom diameter. We have already seen in the figure. The helical structure repeats after every 10 residues. So as we have already seen, this is one helix. Okay, so this helix, it will repeat again. Okay, starting from here, another helix over there. So how many base pairs are here? 10 base pairs. Okay, what is the length? What is the pitch? That is 34 angstrom. So what could be the distance between adjacent base pairs? It is 34 divided by 10, that is 3.4 angstrom. Okay, so it is actually base pairs are arranged 10 base pairs are arranged in a single helix, and it is placed 3.4 angstrom uh, okay, apart. Fine. So when it is in solution, what happens is it is not exactly 10 residues, it is 10.5 residues occur in a single turn of the double helix when it is in solution. Okay. Now the two helices, they are wound in such a way as to produce two interchain spacings, and these interchain spacings are known as grooves. Okay, you may have seen like these coil ropes wound around, isn't it? There will be two strings, and they may be wound around in a helical fashion to form a coil rope. Okay, and this coil rope, if you just unwind it slightly, you can see so many grooves, so many gaps in the at various places. Okay, and these spaces are known as grooves. Okay, if you see the structure over here, so this is a groove. Okay, you can see here, this is a groove. Okay, this is yet another groove. This is this groove and this groove, it is almost similar, and here a smaller one, here also there is a smaller one. Okay, based upon its depth, we can say this is a minor groove and this is a major groove. So in a double stranded DNA, they have two interchain spacings, and these are known as grooves, and based upon the depth and the width, we have a major groove with a depth of 8.5 angstrom and 12 angstrom wide, while a minor groove, which is 7.5 angstrom deeper with 6 angstrom wide width. Okay. Now, what are the features of major groove? Major groove, as we have already seen, it is deeper and it is wider in nature, and obviously it is deeper than the minor groove, and they are the sites for specific proteins to interact with DNA sequences. So what kind of proteins will interact? You may have already studied DNA replication, and during transcription, the DNA getting transcribed to mRNA, isn't it? So at all these occasions, what happens is the DNA has to interact with many different kinds of proteins, and those specific proteins, they bind with major groups. Okay, so the major group provide a platform where the proteins can interact with the DNA sequences. Okay, now what are minor groups for? Minor groups, we can see that small molecules like drugs and all, they bind to the minor group. Okay, now, so what makes the double helix stable? Okay, even how, what makes the DNA double helical, and why it is stable? Uh, you can see here the bases, they are hydrophobic and they are shielded from water because they are spaced internally, and the what you call the uh, two chain backbones, it is found the phosphodiester bonds between two sugars. So sugar and phosphate, they are hydrophilic in nature. The phosphodiester bonds, they are hydrophilic in nature, and this helps in shielding the nitrogenous bases from the aqueous environment. Okay, the watery medium outside. So it becomes more stable, and this helps in protecting the genetic information from any physical and chemical attack. What is the genetic information? The genetic information is stored in the sequence of the nitrogenous bases, because that is what is specific, and that is what is read during the transcription process, and mRNA is formed based upon the template of the bases on the DNA. So bases are actually, the sequence of bases are actually the genetic information. So it has to be protected from physical and chemical attack. And when the bases are placed within the two helix, double helix, the two chains, what happens is it is completely protected. Okay, so hydrophilic phosphodiester bonds, they are exposed to water, they can interact with water, while hydrophobic nitrogenous bases, they are placed inside the two chains inside the double helix, and hence it is protected from the watery medium, so it becomes more stable. Okay, now so this is the structure. You can see, you have to learn to draw the diagram. You can see here, this is one turn of the helix. This is one turn, and the whole turn, it is 34 angstrom in length. Okay, and you can see there are 10 0.5 base pairs, 10 base pairs in a single